Spanish, pero el acentito nunca se quita. What will we do today is about the cell theory. We're going to introduce the cell theory. It's a very simple theory. What is a little bit more complex is how the cell theory came to be. So we're going to see a little bit of history, okay? But before that, the class of today, I would like to start it with a video that we didn't see today. It's the video of the endosymbiotic theory. So I really recommend you to subscribe to this channel, which is called Kurzgesagt, because they have the most amazing animations and really, really great explanations, and they're scientifically very correct. So I really recommend you that you subscribe to this Kurzgesagt because they're very applicable in a lot of our subjects of this course. And today we're going to see a video of this. It takes about 11 minutes, so it's going to take like 1/5 of the class. Then we're going to see what this cell theory is about. And then I would like to finish with a quiz, okay? Don't get scared. I didn't tell you in advance that we're going to do a quiz today, but that's why I also give you two trials. You know, you can try twice. You're going to do once in class. You will see how you do. If it goes bad, no problem. You can do it at home again and get a better grade, okay? So I think that's very fair. My objective is not for you to give you quizzes without studying. It's just to see where you are. I think it's very important to now and then do a quiz in the class without advising, you know? And I do that deliberately because in that way you get confronted with the fact of how much you understand or not, how useful have the classes been for you, or how much did you pay attention. In fact, if you do the quiz today and you get a 40, let's say, then it means that you have to revise things better. You have to pay a little bit more attention to the course, and it means that you have to study a little bit for this part, you know. So it's really a confrontation with where you are at understanding the matter of this subject. So even if you have a bad grade, no problem. You can study whenever you want and you can take the quiz again. So you get a chance for yourself to do it whenever you want, okay? So it's more like a homework quiz just to see where we at, but it does count for points, okay? If you get 100 in the first time, okay, you can leave it like that. If not, then revise the content and do it again whenever you feel ready. However, it has to be within the next week, okay? So first things first, like always, and let's start with this video. So that's a little bit the content of today. So we start with the video. We look at the cell theory, not the cell theory itself and the history. And then we finish up today with a little quiz about the cell organelles, okay? So let's have a look at this. And again, guys, please subscribe to this. I even watch it for fun, you know. Sometimes I say like, this is interesting. Let me watch a video of Kurzgesagt.
I do follow that account.
Yeah, I already followed them.
It's very good.
They're lit. They're lit.
There's
also
some other
that's pretty good.
Yeah, there's some other channels that are really, really nice. They're not especially about biology.
But Kurzgesagt is #1, Mister. They're very good.
Yeah, but some other really good ones that I really like are called Polymatter. It's more about economics and logistics and business and stuff. It's really interesting. However, it's nothing to do with our course. And another one that I really, really like is Atlas Pro. Maybe I share them one day with you. I follow certain channels that are very interesting. So let me share.
Vsauce.
You can also share with me. I am very aware that I can learn things from you guys. Certain students have shown me very interesting things, and I'm very grateful for that. So here we go. Let's start with the video.
At this very second, you are on a narrow ledge between life and death. You probably don't feel it, but there's an incredible amount of activity going on inside you. And this activity can never stop. Picture yourself as a slinky falling down an escalator moving upwards. The falling part represents the self-replicating processes of your cells. The escalator represents the laws of physics driving you forwards. To be alive is to be in motion, but never arriving anywhere. If you reach the top of the escalator, there's no more falling possible, and you are dead forever. Somewhat unsettlingly, the universe wants you to reach the top. How do you avoid that, and why are you alive? All life is based on the cell. A cell is a piece of the dead universe that separated itself from the rest so it could do its own thing for a while. When this separation breaks down, it dies and joins the rest of the dead universe again. Unfortunately, the universe would like for life to be done with doing its own thing. For some reason, it's not a fan of exciting things, but tries to be as boring as possible. We call this principle entropy, and it's a fundamental rule of our universe. It's pretty complicated and counterintuitive, so we'll explain it in detail in another video. For now, all you need to know is living things are inherently exciting. A cell is filled up with millions of proteins and millions more simpler molecules like water. Thousands of complex self-replicating processes are happening up to hundreds of thousands of times every second. To stay alive and exciting, it has to constantly work to keep itself from achieving entropy and becoming boring and dead. The cell has to maintain the separation from the rest of the universe. It's doing this, for example, by keeping the concentration of certain molecules different on the inside and the outside by actively pumping out excess molecules. To do stuff like this, a cell needs energy. Energy is the ability of things in the universe to do work, to move or manipulate a thing, to create change. This ability cannot be created or destroyed. The set amount of energy in the universe will never change. We don't know why, it just is that way. So, billions of years ago, one of the most crucial challenges for the first living beings was to get usable energy. We don't know a lot about the first cells, except that they got their energy from simple chemical reactions. And they found the ultimate energy transfer system, the energetic building block of life, the molecule adenosine triphosphate, or ATP. Its structure makes it uniquely good at accepting and releasing energy. When a cell needs energy, for example to pump out molecules or to repair a broken micromachine, it can break down ATP and use the chemical energy to do work and create change. This is why living beings are able to do stuff. We don't know when or how exactly the first ATP molecule was made on Earth, but every living thing we know uses ATP or something very similar to keep its internal machinery running. It's crucial for almost every process plants, fungi, bacteria, and animals need to survive. Without ATP, no life on Earth, possibly anywhere. While breaking down chemicals for energy is nice and all, early life did miss out on the greatest available source of energy, the sun. The sun merges atoms and radiates photons away that carry energy into the solar system. But this energy is raw and indigestible. It needs to be refined. After hundreds of millions of years of evolution, finally, a cell figured out how to eat the sun. It absorbed radiation and converted much of it into neat little chemical packages that it could use to stay alive. We call this process photosynthesis. You take photons that are wobbly with electromagnetic energy and use a part of this energy to merge and combine different molecules together. The electromagnetic energy is converted into chemical energy stored in the ATP molecule. This process became even better as some cells learned to create better chemical packages. Glucose, or sugar, easy to break down, high in energy, and pretty tasty. This is so convenient that some cells decided that instead of doing all that pesky photosynthesis work themselves, they would just swallow other cells that did and take their glucose and ATP. This is widely considered one of the biggest anime betrayals in evolutionary history. And so things went on. Photosynthesizing cells could mostly harness energy at their surfaces, which limited their maximum energy production, which limited their evolutionary avenues somewhat. So time passed. Some cells made sugar, others ate them. Evolution did its thing, but overall things stayed pretty much the same for hundreds of millions of years. Until one day, a cell ate another and did not kill it. Instead, they became one cell. Nothing had changed that day, but Earth would be different forever. This cell became the ancestor of all animals on this planet. Blue whales, amoeba, dinosaurs, jellyfish, pink fairy armadillos, and sunder colugos. And of course, you. All can trace back their existence to this moment. The merging of two living beings is so important because when those two cells became one, they became way more powerful. The formerly independent cell in the inside could stop trying to survive. It could concentrate on one thing, make ATP. It became the powerhouse of the cell, the first mitochondria. The host cell's job became to ensure survival in the dangerous world and provide the mitochondria with food. Mitochondria basically reverse photosynthesis in a similarly complex process. They take sugar molecules that we got from eating other living things, combust them with oxygen and precursor molecules to make new, energy-rich ATP molecules. This process works like a tiny furnace and spits out waste products like CO2, water, and a little bit of kinetic energy that you experience as body heat. This first division of labor meant the new cell had way more energy available than any cell before, which meant more possibilities for evolution to enable more complex cells. At some point, these cells began to form small groups or communities, which led to multicellular life and finally to you. Today, you are a pile of trillions of cells, each filled with dozens, if not hundreds of little machines that provide you with usable energy to stay alive. If this process is interrupted, even for a few minutes, you die. But if life is so fragile, wouldn't it be a good idea to store ATP, like we store sugar in our fat cells, so we don't die if we stop breathing for a while? If life has solved so many problems to make you live today, what's up with the dying quickly thing? Even simple bacteria like E. coli make about 50 times their body weight in ATP for every cell division. Your trillions of cells need a lot of ATP to keep you around. Every day, your body produces and converts about 90 million billion molecules of ATP, about your own body weight. You need a whole person's worth of ATP just to make it through a single day. Even storing enough ATP to last you a few minutes is basically impossible. An ATP molecule is really good for shifting energy around quickly, but it's terrible for storage since it has only 1% of a glucose molecule's energy at 3 times its mass. So ATP is constantly produced and used up fairly quickly. This was the short and simplified story of the molecule that allows you to be different from the dead universe and to be the slinky on the escalator. It is a weird story. There is this molecule you need to survive at all times. You need it to keep moving because even a short break brings your slinky to a stop. And you need to make it yourself. It's like driving a car at full speed while producing fuel in the trunk with junk that you pick up from the side of the road. As far as we know, this all began billions of years ago when tiny parts of the dead universe came together and became something else for a moment. It could keep itself going. It could grow. That moment set the slinky in motion, and it's been going ever since. From the very first cells to you watching this now. At some point, you will merge with the rest of the dead universe again. Maybe you'll tell it stories about your adventures. Maybe not. But before you find out, you get to do what life does best. Making a dead universe much more interesting. If you are currently bored for whatever reason and you want to make your life more interesting, we've got something for you. We partnered with Skillshare,
Okay. Um, that's it. Isn't it fascinating? So what you see in this video is in fact what I think it's very nice to compare with human beings. So I like to compare the endosymbiotic theory a little bit with us humans. So think about us humans, how we became civilized, let's say. Okay, think about a million the first humans. How did they survive? Well, we were individuals. We would run around in the savannah, and we would try to find food. Okay, that's basically how humans started out. Little by little, we would form little groups, okay, because we would know that humans, we are stronger if we're in little groups. So um, you get little groups that would hunt together, you know. And the same is with the endosymbiotic theory. The cells started to work together, and they could do different functions. This is how humans became civilized and how we became so dominant over other species, is because we work together. Think about the first villages. I mean, if you have a community, a group of people that first was way more individualized. I do think that people always had some kind of family lives, of course, because mammals cannot survive without the care of the mother. But let's simplify things. But think from the moment that we were having villages, and for instance, we would work together, and you would have one person that would take care of security. You would have two other persons that would gather food. You would have another one that would build the houses. You would have somebody else who would do the cooking. You would have somebody else taking care of children. If everybody would get specific tasks, then the benefits for everybody would be greater. The same is with cells, you know. You see that this is not a coincidence. We see it on all levels. And cells did exactly that, you know. And that's what the main ideas of this video is. Not that just two cells, they joined together by accident. No, they joined together because they had a benefit of doing that. One cell could search food, and the other one could produce energy. So there was a motive to 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 join together. There was like a mutual interest that grew in the end all life on planet Earth. And we see that on all levels. Think what I just said about humans, think about cells, think about small animals, think about ants, think about bees. All animals, this is also a thing that we see so often in nature, that animals work together in order to achieve a certain thing. Fish swim in big groups together in the ocean. Birds fly together. Ants work together. Bees work together. Humans work together. Wolves work together. Cells work together, you know? So that's a bit the endosymbiotic theory, you know? And after millions and millions of years, of course, you have energy, too much energy. There is benefits you can do with this, and you can go to other levels. And this is exactly what happened with the cell. So this video, I really, really like it. I think it goes into detail. It explains very clearly what the endosymbiotic theory is about. Does everybody understand that, or do you need some more explanation about this? No, we're
Teacher,
fine.
No, it was
Go ahead.
what is the name of the YouTube channel?
Sorry, the name of what?
The YouTube channel.
It's named Kurzgesagt. It's German for saying gesagt is set and Kurs is Kurs. So it's in German Kurzgesagt.
Mister, I had a question.
Yes.
What was the mitochondria?
The mitochondria is the powerhouse of a cell. It's a small part, a small organelle of the cell, and scientists think that this is like adapted bacteria that is living inside a cell. So that the origin of the mitochondria is a bacteria that was able to produce energy. It fused together with a cell, and it became like the entity, the organelle of the cell that makes sure that the cell has energy to do whatever the cell needs to do. okay? So the mitochondria is really the powerhouse. It takes care of making sure the cell has energy. So let's go to the second part of the course, because otherwise we cannot finish. This is a little bit about how cells were discovered. So we're not really talking about the history of the cell here. We're more talking about the history of discovery, okay? So keep that in mind. Why do we do this? Because it's interesting. It's very interesting to see, to understand how we know what we know, okay? So that's why we show this. So we're gonna talk about certain people. This is the index. We will only get the objectives and the cell theory today. We will not go to the quiz yet. We will do the quiz today of the cell organelles in the canvas. I will go you there in a moment, okay? So how were cells discovered? I hope you can see my screen. Okay, so we are going to see what is a cell theory about and how it came to be. We're going to revise quickly what are the differences between prokaryotic and eukaryotic cell, and we see the differences between the eukaryotic cells, the plant and animal cell. We already did that yesterday, we already did that last week, but it's so important that we go over and over it again, because this is very important basic stuff, you know, that we have to see different times, several times, for you to make sure that that this is well understood, because it's really fundamental to know this in biology, that you know the cell components and its function. So how did the cell theory came to be? Well, in 1595, if you click on it, Hans and Zacharias Janssen, what they did is they made a first kind of compound microscope. Okay, if you click on it, you can see it. It was not really great, guys. They could just see things magnified, but they were not able to distinguish cells. It was not that well done because they couldn't make the glass clear enough. It would have a lot of distortion of light. It would have a blurry look. So they could see things a bit bigger, but really they could not see cells or really details. Moreover, they had no clue what they were looking at, but anyway, it was the first thing to what we can call a microscope today. And then, almost 100 years later, Robert Hooke, well, he did something different. He improved a microscope a little bit. He made a better one. What he did, he was able to polish the glass in a better way, so they had less light distortion. And he was also in a monastery, so it was a kind of monk. He had a lot of time, and I suppose he was kind of bored. So what he did, he made this microscope, he put a piece of cork on it, and in fact, this is an important person, Hooke, because he is the person that said a cell is a cell. And where does the name come from? When he was looking in the microscope, think about the onion you have seen in the virtual microscope. He was looking at these little circular rectangular things, and he was thinking like, oh, that's weird. It looks exactly like the little room I'm in. And the rooms of monasteries were called cells. So he said that cells should be named cells because it looked the same as his room. Okay, that's where the name cell comes from. So he is important because he did improve the microscope. He must have seen something like you see here, and he could see all the little squares. And he was thinking that life was made out of little rooms as well. So that's why he came up with the first time that a cell was a cell. Okay. Not that much later, Anton van Leeuwenhoek, a Dutch person, my father is Dutch, so it was in the Netherlands that this was done. He really improved the microscope even better. And what is really important, what van Leeuwenhoek did, is that his microscope didn't look like the previous one. What he did, he just had a piece of wood and he had a piece of glass. He polished so nicely that he could look through the eye like a loop, you know? And he looked they didn't brush the teeth so often those days. So he got out of his mouth some yellow stuff on his teeth, which is full of bacteria. He put it on a piece of wood, or I don't know where he put it on, but he was looking at it at like the mugrero, para decirlo así, of his teeth. he looked at it and he saw that things were moving, and he was a little bit shocked by it because what he actually saw were like bacteria, you know. His microscope was strong enough to see bacteria. So he said, like in 1647 and 1674, that there were little animals in the scrapings of his mouth. We all have that. I mean, we should have less because we do brush our teeth. But in those days, yeah, the mouth was a place of infection, and it was full of bacteria because they didn't have the same hygienic standards as we have, and didn't disinfect. So he could see a lot of things moving. In fact, if you would do that today, you would also see things moving. I don't think you don't have bacteria in your mouth, because we all have. Okay, but he called it animacules. He thought like there were little animals, so he called it animacules. Okay. Then there was Schleiden and Schwann. What they did is they heard about Van Leeuwenhoek and what he saw, but you have to remember that in those days you could not say things so aloud, you know. If you would write down these things and you'd say, yeah, I've seen little animals in my mouth, and you would write it, then it would be against the church and people would say you were crazy. But he got the attention of certain scientists, Schleiden and Schwann. So they investigated a little bit more, and what they saw is that both animals and plants, they seem to be made of exactly the same structure and of cells. So they looked into different things. They were investigating, and they would look at animals, they would look at plants, and they would see every time the same thing. They would always see like similar structures, the cells. So they came up with the fact that everything was made of cells. And then we have Virchow, 20 years later, who we can call the father of the cell theory, because he is the first one that defined that all cells come from other cells. So why is this important, these people? Because those people, if you go through history, all of them, they made the cell theory. And the cell theory is something very simple. You have to know it, but it's 3 big rules, okay? The first one is that all living things are made out of 1 or more cells. So this is something that Schleiden and Schwann discovered. Then the second thing is the cell is the basic unit of all living organisms. This is an idea that all of them had from the first moment that this Hook came up with the name cell, you know, that all life is the basic unit of cells. And all cells come from pre-existing cells, so that all cells are born out of other cells. And this is in fact Virchow who really defined this. So this cell theory comes from different kinds of scientists that came to different kinds of conclusions, and that this is in fact the law of the cells. Very simple. All life is made out of cells, okay? The cell is the basic unit of life, so it means it's the smallest thing that can be alive is a cell, okay? Don't go to viruses yet, because that's the discussion area, but the smallest thing alive, the basic unit of life is a cell, because it's like a isolated piece in the environment that can maintain and reproduce itself. And there you go with the characteristics of life that we have seen before. And the third law is basically that all cells come from other cells, okay? So now I go a little bit fast because we already did this. This is the bacteria we have seen, okay? The cytoplasm. Plasmids is like the little piece of DNA that you have in the bacteria. They use it to infect other bacteria to have genetic variety. There you have the DNA. You have a plasma membrane. All cell membrane and plasma membrane is very similar to thing. The capsule, certain bacteria, they have a kind of protective membrane around it, can be made of sugar, can be made of certain proteins. It's like a skeleton, an exoskeleton, you could say. Some bacteria, they have little hairs, you know, we call them cilia, and the other one, cilia or pili. Pili is when it's used to put plastids. In flagella is when it's longer, okay? So maybe we should go a little bit more in detail. Pili and cilia, there is 2 things. Pili is used for reproduction, okay? and cili is more for movement. Okay, so the pili is when the plasmid goes through the pili through another bacteria, while cili it's more for movement. Think about amoeba who has like little hairs to move around. It's not so important, guys. Okay. Okay, eukaryotic cells. This we did yesterday, so I'm just going to skip it. Otherwise, we don't have the time to do the quiz. But remember the cell wall, the cytoskeleton, the mitochondria, the plasma. This we don't. It's plasmodesma is to connect cells to each other. Plasma membrane. Lysosome, we have seen that. Golgi body, we have seen that. The smooth ER, the rough ER. We have the ribosomes, the nucleolus, the nucleus, with the chromatin, nucleolus, chromatin, and DNA. Okay. Vacuole, chloroplast. So here, what is the only thing new here is the plasmodesma. Okay. So cells connect to each other. We call them desmosomas or plasmodesmas. Depends what the connection is. It's not that important so at this point, but for instance, if you have skin, your cells in your skin, they're connected. That's why when I can do this, my skin comes up. It's because all the cells are connected to each other with desmosomas, we call them, desmosomes. In plant cells, they call plasmodesmas. It goes with different names. It depends the connection, but it's not that important for now. Okay, here you have the eukaryotic cell. Okay, here this is the centrioles. They make microtubulus. Okay, we have a cytoskeleton. We go see that later in more detail in the next chapter. Okay, but a cell can have a skeleton inside. They needed to transport things, to divide, but we will see that in cell transport and in mitosis in more detail. So the rest is things we have already seen. Okay. Okay, here you have a summary of this.
Mr.
Yes.
What type was the blue cell? What type of cell?
It's you have here the plant cell, okay? With the differences is the cell wall, the vacuole, and the chloroplast. And this is an animal cell. The next one is an animal cell. So it has no cell wall, it has no vacuole, it has no chloroplast. Okay, so that's the thing you can easily recognize because here you see typical animal cell components.
Okay.
So check also in iCell in your application, you can see those differences. I'm just repeating myself. I'm not saying anything new except for the connection. This is a little bit of summary, what you have in all kinds of cells. Okay, there's nothing new. And here we are gonna leave it. The next thing we do tomorrow. And the last 10 minutes, I would like you to go to this quiz and adjust to see where we are. Because for me, it's important that you understand everything. So that's why I want to do this. And don't be afraid, guys, you have 2 trials. I give you 2 trials. So if you don't do well now, you can always try again after revising everything in detail for yourself tomorrow or later. Tomorrow, by the way, we do a little quiz for real that you have only 1 trial about the endosymbiotic theory because that part is finished, but this is a trial. So please go to Canvas and then have a look here into week number 7, sorry. And go to cell types and their organelles. Okay, this one here, review questionnaire. And please do this quiz. And that's the last thing we do today. It's a very short quiz. It has a few questions, but it's just to see how we are doing and if I need to give you more explanation about everything. So you get 10-15 minutes to do this. It has a few questions. Please answer them, and I'll see how you're doing. Okay. So don't confuse the quiz. Endosymbiotic theory is for tomorrow. Now you do cell organelles and cell theory, okay?
I only have.
Teacher, I only have until week 6.
Yeah, we'll see. Week
Okay,
6.
don't worry. Let me make it.
Me too.
You have only okay. Let me make it. Yeah, it should be visible right now. It was not published. If you don't see it immediately, you should refresh your canvas quickly, but it should be there now. Yeah, the second partial was not activated yet.
So which activity was it?
It's called Cell Types and Their Organelles Review Questionnaire. This
Okay, got it.
one you're gonna do. Okay? And again, you have 2 trials, so really don't get stressed if it doesn't go okay yet. The objective is just to know if you understand what we have seen or not. If you do bad, you review the content, you study it, and you can do the quiz again whenever you want, okay? So it's just to see if we're doing fine or not. It's like a thermometer, you know? So you only have 4 questions, so it should be okay. It's not that much. Everybody gets different questions. And I'm sure some of you will have 100 because it's not that difficult.
Teacher, I went for flowers. So what are we doing? You're muted. Oh, teacher, do you have your mic muted?
We are doing the review questionnaires of cell types. ¿Qué pasó? Ay, algo prendió. Bueno, we are doing the review questionnaire of cell types and their organelles. But it's almost finished the class, and I think you have another class after. If you don't have time to do it right now, you have 2 trials, 2 intents, so you can do them whenever you want, okay? But you will not have the same question in each intent, okay? The idea is in the first intent is to see how you're doing, what do you know right now? Then if you do well, which I think for most of you will be the case, fine. Then you don't study too much anymore. And if you did bad, then please review the content, study it a little bit, and then do your second trial whenever you feel ready, okay? That's the idea of this quiz.
Okay, thank you.
Okay. In the second trial, guys, you can use iCell at the same moment, okay? Use the application, do it, because by researching, by looking for it, and then answering it, you learn. It's a very good way to learn. So even now, if you want, you're allowed to use the iCell app if you want, okay? Because I see it more than an exercise than really a quiz. However, you do get points for it. And the highest grade will be saved, okay? So whenever you want to do the second one, you can do it. If you don't want to do it, also fine. But I do recommend you do it because similar questions, they come out of the same database when we have the second partial. And remember that we have a little bit of early second partial this year. Normally it's in the end of October, the beginning of November even. And now it is 15th or something. I don't remember the exact date, but it's fast. It's really fast. I think, yeah, I think it was the 15th. I'm not exactly sure, but I do think it was the 15th. It should be somewhere in my announcements. In fact, if you want, I can revise that to be sure. So there is no doubt about this. I have so many dates I have to remember that I really have to check to be sure, you know. I tell you in a second, wait. All right. Yeah, second partial is on the 15th of October. I'm going to check it with the academic calendar because in my calendar I put it on the 15th. So let me double check with the academic calendar to be 100% sure. And then later on today, I guess I will spam you with some gifts I have about the cell organelles. Keep them. It's nice because they're like funny to see. And when you look at them, it's also
Sure, I got a 19.
19. Okay, then you have to study a bit.
Without using the iCell.
It's not
90.
90.
No, like 19. 90.
90. Okay, I thought you said.
Yeah.
No, 90 could not be so good, but 90 is fine. It's fine. Then you just missed some.
No.
Well done, Emilio.
Thanks.
So we also don't know if we go back to campus yet. There's still a lot of doubt according to this. In Europe, things are going really bad with coronavirus. Mexico, I guess it will follow very soon as well, more cases. If you ask me, I really don't know what the outcome will be now. The whole world is trying for 6 months to contain the virus. It seems that we're not able to do it. So I think we should find a way to live with it instead of locking us up all the time. So there must be other ways, you know. I think we have to try something else. So yes, guys, it's on Thursday, the 15th of October. And if we go back to campus, it will be divided over 2 days, but we're not there yet. In fact, I'm not sure if we will go back this semester. And be careful, I don't know what will happen next semester either. It's they don't know yet. But I do think we have to find a solution somehow because we cannot stay online forever, you know. But I'll keep you up to date. We honestly don't know. I think tech is defining certain things. They always wait a little bit with bringing the news out because they want to avoid speculation, which I think it's fine. You should be careful with what you say because otherwise people have only doubts. I can only share my opinion with you guys. So if you're ready, guys, you can disconnect. You don't have to wait. I know you have another class. Sorry that we went a little bit out of time now. Yeah, do the second chance on your own whenever you feel ready for it. Okay. I see some very good grades already.
Goodbye, teacher. Have a nice day.
Bye. Have a nice day too.
Bye, teacher.
Thank you.
Sure. My quiz didn't load
Bye bye.
because of my internet. May I make it
Sure, Jose, I'll do it whenever
in the afternoon?
Yeah, of course. Don't worry. Just make sure you do it before the expiry date. You can see when you have to finish it. So, okay. And see you tomorrow. Take
Thank
care.
you. Bye.